Here we are again. And so Riley got sick, you guys, and he's sorry he can't be here, so we're going to hope that he gets here on Tuesday, and it would be really good timing because he wants to talk about the cellular response. And that's really what Chapter 18 is. It's an example of a cellular response, and it's just our favorite because this course is informally subtitled Mechanisms of Cancer. And so the troublesome response in cancer is primarily the cell cycle gets going too much. So that's a cellular response. It's happening under the scab. The skin cells are making a cellular response to PDGF. And in that case, it's to go through that start and go through the cell cycle like that. And so Riley wants to talk about that. That's one thing that the T cells do when they're activated to kill the cancer. One thing they do is they proliferate so that more of them will be present to do more fighting, both now and in the future, like that. And but this T cells make some other responses too that he wants to talk about. So that'll be good. We'll hear about that. Then we're reading chapter 18. That's our specific one, cell cycle, and it has that double value to us. It is a cellular response like that, and the response is very significant in molecular biology because of the orthologs that are involved in regulating the cell cycle, and the fact that we can actually describe specific pathways like this domino, it's that domino, it's that domino. And I know that some people had kind of a sort of an epiphany when they're like, oh my God, on Tuesday, where you're like, oh, PDGF binds to the PDGF receptor. PDGF receptor autophosphorylates and activates RAS. RAS has GTP, it's active, it activates RAF kinase. RAF kinase activates phosphorylates and activates MEK, and that phosphorylates and activates ERK, and that phosphorylates and activates ELK, and then ELK is a transcription factor that binds the enhancer of a lot of target genes, including a cyclin. And the skin cells are waiting at start in the cell cycle because the green light is caused by an enzyme called cyclin-dependent kinase. And it's waiting, the light is red, in this case, just because there's not enough cyclin. So what's the role of ELK? ELK helps make more cyclin like that. And some people had an epiphany because that's what molecular biology is like, is you're just like, oh my gosh, really? There's no step where we have to just go, and then, it's too advanced, it's too technical, just pretend it's magic. No, cool. Now, the truth is, we'll talk about it some because ELK is not an oncogene. It's not a famous oncogene. Another player in other cells on that similar pathway is MYC, and MYC is a big-time oncogene. So there's more to it, but the framework is solid, and it's just a question of it going out further and expanding out further into that chapter and into the next couple of chapters, like that, okay? Because we'll see that when, say, MYC activates cyclin synthesis, because MYC can do that. MYC also activates synthesis of other proteins that say to the cell, maybe you should just die. And that's actually chapter 19, because one thing that really often happens is that when a cell is stimulated to go through start, it's also encouraged to decide whether or not it's doing the right thing. And if not, it should go through chapter 19, programmed cell death, which is also called apoptosis a lot of times, and you may have heard of that. So it gets complicated. We just have to add more knowledge. But there's this framework that's just a rock and roll framework where you're like, okay, that's good. So when I do that, chapter 18, we're going to keep going on 18, and we won't finish it today, but we're doing it great. So we want to start looking at chapter 19. Chapter 19 is a really important chapter also, but it's a shorter kind of chapter. It's not as meaty as chapter 17 and 18 have been. And so, and part of that is because it's about stem cells, and we've already talked some about stem cells, and stem cell biology is pretty straightforward in a course. Okay, so that's their flow, and I got some of these out, but I also left some in just to to talk about what we talked about. So our overarching picture in our course could be said, what happens under a scab like that? It could be that, and it's a story of regulation of the cellular response in skin. So it really starts with this regulation by cell signaling. The PDGF binds and stimulates the cell cycle. And it does that through a pathway of new gene expression and it leads to a cellular response, and in this case, it's that cell cycle, the focus of chapter 18. And so, although we talked about them in this order in our course, the story is really that's going to first be the cell signal, then a signal transduction, and then maybe a new gene expression, and then some kind of cellular response like that. And we just have to be rock solid on that part of our course because we've talked about it so many times in class, and of course the book covers it as well, but we just have to be rock solid on that because that's just our foundational framework that we stand up on and to learn more about. And so we've shown that before, like that, okay? And then we add this vision of it, and that is that this, in our story, this pathway leads to regulation of that start. And we can say start is like it's a control point. It's like the button on the washing machine that the PDGF presses to get the whole thing started. So start's a great name. It's also called the G1 to S phase transition. People always talk about it as start as being here in G1. It's definitely in G1. Realistically, it's just before S phase because we'll see our textbook talks about it. So it's weird that people continue to talk about G1 at the start as if it's like some time before S phase. And we'll talk about it. It's actually because I mentioned last time that people who are really into this call this a control point as a general term, and that's to distinguish it from other checkpoints. And checkpoints are when the cell looks internally. Is there DNA damage? Are the chromosomes getting separated properly? And a control point is where it looks outside. It says, I'm just waiting to think about, should I go forward, like that. And there's lots of interesting things to say about control points because every eukaryotic cell on our planet has that start. That control point is waiting for external signals like that. In the case of the yeast that causes bread to rise, they're not waiting for a hormone signal or a paracrine signal. They're not waiting for some external signal because it's just yeast. Single-celled eukaryotes aren't waiting for some other team member to say, hey, go. What they're waiting for, the yeast is waiting for, is a nutrient signal. So, there's considerable diversity in the regulation of start, but the actual players, every eukaryote has a ortholog of the CDK, the cyclin-dependent kinase. And when it gets activated, it just starts phosphorylating stuff that's needed to synthesize new DNA. And so our book describes it like this, right? Here's this flow, and it says there's your pathway, there's your synthesis of the cyclins, and then you got the complexes together, and they can, that's what is the light turning green. I like this visual. I always like visuals like that. It's the same thing. And our book shows this a couple of times. So that's the story of the woman with skin cancer. She's got just that pathway, and she's autocrine stimulating her own skin cells to grow fast. So in chapter 18, we have these three parts we want to chat through. Most of our emphasis is on start, and so we'll go today and talk about some more examples of start. But start is really complicated because there are lots of checks and balances like that. And so we'll learn a little bit about the checks and balances, but before we do that and come back to talking about start, we'll shift over to another point in the cell cycle called a DNA damage checkpoint at the G2 to M transition going into mitosis, because that one is really understandable. It's really simple. The cell's going to wait, like the wash machine with the lid up. They'll wait there all day until what? Until the DNA damage gets corrected, and that's like putting the lid down, and now it can go into mitosis. And then the book talks about, and we want to spend a little bit of time on the phases on mitosis, what you see under a microscope and what's happening with the chromosomes during that M phase. Okay? So there's that PDGF pushing the button like that. That's a growth factor, platelet-derived growth factor, great name for it. Pushing the start like that, making the light green, and we know about how it does that. So I there, again, showed these last time. There are those three steps in the cell cycle that we're learning some about. We're already almost done talking about that one. Then we'll get over to this one and have a nice little package of understanding about that one, the DNA damage checkpoint at the G2 to M transition. And we're going to talk here more today, and then we'll get back to that because it's everyone's favorite, because that's the switch. When a cell starts, when a cell goes through start, it almost invariably goes around and stops again. It will pause, like the washing machine, but it will go around again, like that. And so that's why it's called start, because it's where they're gonna wait, like that. If you looked at our normal cells, you know, and you say, look at 100 skin cells, almost all of them are at start. And only a small percentage have gone through start, because you know our normal skin has to slowly grow through the cell cycle to replenish our skin, right? Always getting rubbed off, dead skin cells, always gotta replenish. And that's true about our skin cells, so it's relevant for that person with skin cancer, right? Because she's taken that normal regulatory pathway and activating it like we need to hurry up because it's under a scab, she thinks, like that, right? Okay, so when you look at cells, like skin cells, they're mostly just waiting there. And so you'd say that skin cells are slowly going through the cell cycle. What that's like is, it just means that they have to sit at start for a random but long length of time, and then they get a green light and they go through. So in our normal skin, there's always a small percentage of cells that are just scattered through other parts of the cell cycle. At any point, there are some skin cells right here. Some of these skin cells are going through mitosis right now, but just a very small number, because most of the skin cells in that area are just sitting at the red light. Yep?
I saw another source naming it a commitment point. Is that the same thing as a checkpoint and control point?
Well, yeah, so I'll say probably. I don't really know the use of that term. Commitment point. Like, is there a context for that, do you know?
When the cell, I mean, it's on all the same areas of the cell cycle, but I think it just wants to signify that the cell is committing to the next phase in cycle.
Yeah. So sometimes, yeah, because I think that the person is probably using that word commitment in a larger setting where the cell is going to do some events that include the cell cycle. Like we learned about muscle differentiation, like that. And so when the cell is going to decide to go through muscle differentiation, a lot of times cells go through something called a commitment, which is kind of a loose term, not a really technical, detailed term. And in those cases, sometimes that involves the cell going through the cell cycle. In other cases, it involves the cell stopping the cell cycle. So yeah, but I think that's probably where they're used in that kind of context, where there's a bigger meaning that includes cell cycle.
Like this one says SJ's entry commitment point, or mitotic entry commitment point, or mitotic exit.
Is that
point.
out of our textbook or a website?
No, it's a separate source.
Yeah, and so I would say that they're, well, I'll just say that's garbage. Hey, that's garbage. That's what is that? Misinformation. There you go.
Okay, so we're going to use
It's
control
like
point,
some-
checkpoint.
Yeah, sure, yeah, definitely. Definitely that. I think most of the time somebody would say, oh, it's some commitment thing. They're talking about a bigger picture. And if somebody's saying, oh, it's a commitment point, it's that. Yeah, that's probably people who, I don't know, they're, yeah. I'm not, yeah, that's not, definitely not a common term.
Okay.
Yeah, so, yeah.
For the shaky washing machine at M checkpoint, what's the comparison of that?
Yeah, so that's where, in a really way that you're familiar with, the chromosomes get lined up during metaphase, and you gotta separate them. You gotta pull them to the opposite poles using the mitotic spindle. We got microtubules tugging on them, and then they separate. And they gotta get to two opposite poles because we're gonna take this one cell and we're gonna divide it in two. So let's get these chromosomes evenly separated. And there's some bookkeeping that has to go on there. Are we doing that right? Are we getting each of the chromosomes lined up and separated? Or is one kind of going with both of its partners over to the one end? And so people call that the M phase checkpoint for not just mitosis, but also metaphase, because that's when it's happening, and it's like the cell has ways of checking like that, of checking to see if that's right. And other people call it the mitotic spindle checkpoint, because that's who's pulling, the mitotic spindle is pulling on the chromosome, so it's kind of, that's who's doing the work. And what's happening there is just that it can be defective, and as a result, chaotic, where chromosomes don't separate fairly, like that. And I don't think I mentioned it last time, but When you have, like a person has kind of advanced stage cancers, really commonly in advanced stage cancers, when you look under the microscope, the cell has just the wrong number of chromosomes. I mean, it's just chaos. And the word for that when a cell has the wrong number of chromosomes is aneuploid. And I'd say most advanced stage cancers are aneuploid. And it's just because the cancer is growing and it's out of control, and it's just, sometimes that can be helpful when a cancer growing out of control if it's just got mucked up its number of chromosomes. Usually that's really problematic for a cell. Oh, there's a little issue here. And so that's what that is. And that's all, we're actually kind of done talking about that. I don't know why this used to be so good at connecting by itself to Wi-Fi. All right. Oh, no, that's. Huh. Okay, sorry, I had to do this on Tuesday, so I'll try to remember to get that straightened out. Okay, so that's that. And so that's what we're talking about, and it would be in here that there's this checkpoint to say, are we doing that right? And if not, it stops and it tries to correct it, and it just waits. And in a advanced stage cancer cell, it just doesn't wait. It's just like, whatever, I'm busy causing trouble. And so let's just look through this. It's just from another book, but I kind of like it because it emphasizes these 3 points that we're interested in, and we'll just look at it. It has more detail than we really need to know about some of them, and less detail about others. But start out with just to talk around what do we mean when we say G1S, G2M? Okay, so G1, it originally stood for a gap because it's a gap between the two famous important stages, and that is M phase when the cell divides and S phase when the DNA gets duplicated. So it's just a gap, G1. But it's also true that almost all our cells are in G1, and almost all eukaryote cells spend almost all the time in G1. That's when the cell is being itself. All our brain cells, they've stopped growing and dividing. They're in G1, like that. So it's called gap, but that's kind of an embarrassing name because that's when they do their work, right? The focus here is on regulating the cell cycle, right? And so it's not too surprising that the scientists who focus on the cell cycle consider that G1 is kind of boring, even though that's when the cell does its gene expression and makes insulin or hemoglobin and stuff like that, right? So G1, our cells spend most of the time in G1. And some cells are in G1 for so long that scientists love to call this separate time in G1, they like to call it G0. Okay, so I'll just draw that, take a minute to draw that out. It's represented in our book. And so lots of people draw this cell cycle like this. So here's G1, S, G2, M. And there is start. And we'll put it there just for fun. And some cells spend so much time in G1 that people like to say, oh, there's kind of another sort of detour part of the cell cycle called G0. And they say that because, for example, differentiated cells like muscle cells or like our brain cells, they're going to be in G0 for the rest of your life. They're going to divide again. Then other cells may not divide for a really long time. For example, when we were children, we had cells that were waiting until puberty for estrogen or testosterone to stimulate their cell cycle, because that's one thing that happens in puberty is certain kinds of growth are stimulated that's different than juvenile child growth, right? Like that. And so we have some cells that were just waiting all the time in G1 when we were kids for years and years, just waiting for those hormones, right? And that's just surprising. So people are like, let's call that G0. If a cell really is pausing and spending a lot of time in G1, whether it's a permanent arrest, like in our brain, or a temporary, but maybe many year-long arrest, like in secondary sex cells in our vertebrate body, like that, let's call that G0. And that's cool, that's great. And it's just to underscore that, right? And that's what's going on. Right? What's really stunning to hear, though, is there's no such thing. That's a detour. That's the car getting to go forward on another path. In our brain cells, the car's just waiting at a red light. It's just waiting. It's ready to go. It's just waiting. And people just can't believe that. You know, it's just like, wow. You know, all through our childhood, a cell is just sitting there. But it's just like that washing machine that's at your home right now, or at your apartment, or at the laundromat, just waiting for somebody to come in and push start. Just wait there forever, you know, like that. So it's true, and we'll use that terminology and that logic and reasoning, but the reality, the biochemical reality, is it's just waiting. Cyclin-dependent kinases are inactive. And if they would get activated, the cell would just rock and roll right through the cell cycle, right? Like that. Okay? Okay, so great.
So could you put like cyclin in a neuron to make it divide in like an adult?
Yes, but we've just learned one rate limiting step on that CDK, and there are others, multiple. So the protein's there, but a lot of times it's shut down in multiple pathways. But yes, in print, you know, it kind of in theory, yep, just add some of that and we go, like that, right? So that's G1. GAP1, it's a kind of a silly name because lots of stuff happens, including G0. So what are the things that happen in G1? Well, gene expression, like that, you know, insulin and all these things get made during G1. The cell is ready to go, you know, and do stuff. Then another thing that happens in G1 is start. It's definitely in G1. So start is in G1. And then G0 could be part of G1. So those are kind of our list of things that happen in G1. Then S phase. And S phase stands for synthesis. It's DNA synthesis. It's DNA replication. It's when the chromosomes get duplicated. The chromosomes are invisible at this time. So it's a very subtle process. Under a microscope, can't be seen. But it happens, and it happens there, just in the, you know, kind of out in the middle of the cell's process, copies its DNA. Then G2 stands for GAP2, and in our body, nothing really happens too much in G2 like that. Cells spend some time there, but not too much time there in human cells and vertebrate cells. There's some invertebrates who actually have cells that spend a really long time in G2, and they just do their living in G2 a lot, just doing their thing, and you know, they've duplicated their DNA and so forth, like that. Okay, like that. And the thing for us that's important is that it's really in G2, at the end of G2, where we have that G2 to M transition, which we could also call a DNA damage checkpoint. Because the cell is going to stop before going to mitosis and wait if the DNA is damaged. And it'll wait and hope the DNA can get corrected. It wants to do that before the cells divide, because once the cell's divided, it can't really use the other DNA as a reference to repair the DNA. So we really want to get that corrected before we divide the cells. And so that's what, you know, we want to look and understand about the cell cycle in G2. And then M phase is where the chromosomes condense, so they become visible, and we can watch them. They get organized, they line up, they separate, like that, and we'll learn some about that. And so that's mitosis. And so those are the kind of hallmark features of those steps in the cell cycle. And then we have these checkpoints. And so again, G1 checkpoint, you can call it checkpoint. You can call it commitment point. I apologize, Eva, but you can call it commitment point. And I'll be like, great, good job. And the best name is control point to make it distinct from these other checkpoints like that. And we'll learn a lot about that start. And so let's just look at this list, because, you know, why would a cell go through start? So it says nutrients are sufficient. So that's a nod to these single-celled eukaryotes. They wait at start, right? The yeast in your refrigerator, those little yeast in the jar or pack in the refrigerator, all those cells are sitting at start because they don't have nutrients. And when you make bread, you got some water and some sugar, the nutrients they need to be like, woohoo, we're going for it, right? You gotta have that sugar to get the cells to go through, so they need nutrients. It's just interesting to note that multicellular plants and animals don't rely on direct nutrients to regulate the cell cycle, right? We didn't say that nutrients regulate the cell cycle under a scab. It's always regulatory molecules like hormones or paracrine factors, like that. And so that's down here. So growth factors, right? And then there's some internal things, cell size, DNA is undamaged. And then we're going to get back to that DNA is undamaged because I would say the real reason that people actually make a little space there is because in addition to this external checkpoint requirement, the cell also has a DNA damage checkpoint there. And that gets complicated and stuff, but it's really part of START. And the way to think about it is the red light is red, and you get the cyclins made like that, and the CDK gets activated. But there's a couple of regulators that can actually stop that still.
So it's kind
And
of like in the middle of it? Or
it's like, it's just all, they're all together. The good way to think about it, is the control point is a red light, green light, and your car is pulled up and you're waiting at it, and the light turns green. But right then, one of these school control people come out, and they're a DNA damage checkpoint like that. And they're like, we can't go because there's some DNA damage in the intersection. Like that, right? It being kids. The kids are a danger, and you have to stop until they pull back. The light was green, come on. Get out of the way, you kids. Like that, right? Like that. So this is why START is a little bit complicated. We won't really deal with that much until chapter 19, because one of the things that happens is when the school patrol is out, the waiting car, the person could just turn it off and leave. And what that means is it could just kill itself. If the light is green and the school patrol is out, DNA damage has occurred, you have to fix it pretty quickly or the cell will go through programmed cell death. Like that. It's like I'm willing to wait, but there's some issue here. Like that. Yep. And so just a little taste of that because it's mentioned here. Again, it's one of the things that makes START complicated, but we'll get some pieces and add some pieces. Today we're just focusing on that green light. Okay? Okay, great. Okay, so let's talk some more about regulation of START. There it is, talked about in our book because it was famously really figured out in single-celled yeast, so that's what's represented there. The yeast that causes bread to rise is the name of the species, genus species is Saccharomyces cerevisiae, and it's just a really famous model organism like that. It divides by budding, so that's why they're showing it there. It just, it goes through mitosis, but it has this, kind of unusual cell division where it's making buds like that. Just, and I mentioned last time, all these people are still alive and they're just really old, and some of them have Nobel Prizes, so the author's kind of obligated to have a figure in there about yeast because it's just like, holy smoke. This guy in Seattle won for looking at yeast that causes bread to rise. These two guys in England won. One of them studied the yeast that's often used in brewing beer, like that. So obviously, before they won Nobel Prizes, they were just losers, right? It's like, okay, you're studying beer yeast. Okay, what are you working on? You making beer? And they're like, no, not us. No, we're just looking at the biochemistry of how the yeast grow. None of their families were impressed. They're like, you want a Nobel Prize? Oh my God, you're such a failure. Okay, so, and we talked through this last time, just how that language is just boom, boom, boom in this pathway, regulating that. We get the cyclins made and the CDK needs it and goes forward. Again, looking ahead of the course a little bit, unless the school patrol is out, unless there's DNA damage checkpoint there. So let's just look at this now. So here's our pathway, growth factor, RAS, RAFMAC, ERK, ELK, here. And now this is showing in chapter 20, we're not there yet, but this is showing not the cyclins, it's showing another target gene. So ELK, there's lots of molecules of ELK that get phosphorylated. It binds to all of the enhancers of all the target genes. One of them is cyclin. Here's another one we'll need to know, and it's a very famous oncogene called FOS. And FOS is a transcription factor. So things get a little bit complicated at start. How? Well, we learned that straight shot. Elk could bind and activate cyclins, and cyclins get made and the light turns green. That happens in some cells, that directly. In lots of cells, the targets are other transcription factors. So it's just another step. You know, we haven't made the cyclins yet or some other factors we need. First, we're going to make a transcription factor, and that will add a step. Remember back, we had MyoD, it activated myogenin, but myogenin was just another transcription factor that activated target genes. So we just have to add that. That's one of the complicated things. Yeah?
So do they not require CDK or the
They do.
in addition?
Yeah, the way it is, it's a pathway, and we're just saying this domino sometimes requires 2 dominoes. Like that, you know, it's in the middle of the pathway, and we're just like, it gets a little bit complicated here. And, but it really wants to show you this in chapter 20, because FOSS is here, and this is the first time I mentioned that the author likes to make these little exploding yellow things, and we haven't been commenting on those, but those are famous oncogenes. So, RAS, watch out, you know. The ERB receptor in this figure, right? And FOSS, like that, right? But notice, not elk. Elk is on the pathway, but not. What's going on there? Why not elk? And the answer is just that, just the way some genes are organized, they're more likely, when they are randomly mutated, to lead to trouble. And we saw that, one reason we talk about RAS is it's the most common mutation in human cancers, and we know why. It's because it has a kind of Achilles heel. It is an enzyme that needs to turn itself off. And if you have random mutations, you could damage that turning off. That can lead to trouble, because random mutations, they're just random, and most of the time, they don't help the activity, they decrease the activity. So elk isn't considered an oncogene. It's not very often mutated in cancer. It's on a growth control pathway, but apparently it's just not easy to randomly mutate it and have it cause trouble. Like that. We saw this with ERB, right? ERB is a tyrosine kinase receptor. And it can have this thing where it gets, the coding region gets shortened, it gets truncated, and we lose part of the extracellular domain of the protein because the coding region got truncated, and that causes ERB to just go out of control. Why didn't we talk about that with PGF receptor? And the answer is, well, apparently it just doesn't happen with PGF receptor. That if you get a random mutation in it, you truncate PGF receptor, it's just broken, like that. So it's important to see that's on this pathway, right, there are all these dominoes, but some of the dominoes for some reason are the most dangerous. And how do we know they're most dangerous? Because they're seen in cancer. Oh, mutations in these are common in cancer. And it just must be that if we looked into the details, we'd find some reasons why. Oh, geez, that's unfortunate about how the RAS is organized to function, is, it needs to turn itself off, and that's something that's vulnerable to random mutation like that. So FOSS must have some of those features, and we don't need to know, but just that logic is satisfying to see that. Those are oncogenes on a pathway of growth control. Normal growth control can get disrupted, but there are certain points along the pathway that are especially sensitive, like that, okay? Okay, so that's FOSS, and we're talking about chapter 18, but we're gonna get to 20 before the second exam, so we can foreshadow into 20 and see that now, right? START is complicated because sometimes there are multiple steps within that pathway getting that red light green, okay? And here it is shown here too, and that is that FOSS is a transcription factor, and we haven't seen this yet really, but FOSS binds together with June. So you've heard before that some proteins need to be dimers to work together, and this is called a heterodimer, like that. Just need both the proteins to bind. And you can see why. The way they bind to the DNA, it needs both. So it's like this, where you're gonna bind to the DNA like this, and you're just like, look, I need both my hands. I need both June and FOSS to bind to the DNA well. And that in itself is kind of a regulatory thing, right? Because if you just make June, right? You need June and Phos to team up to grab hold. And where do they grab hold? Oh, they're it's an enhancer upstream of cyclin. You know? So it's like Ellie said, well, how does this fit in? And it's just like another step in a pathway. And the regulatory region of cyclin, a good test question would be, what are the enhancers that we've learned about? Well, there's an enhancer that could bind elk. And here's another enhancer that would bind June Phos, like that. There's another enhancer that could bind Mick. And that's true of a lot of genes, you know, that we haven't really talked about, and that is lots of genes have multiple different enhancers and could be activated in different ways, in different cells, different stories, like that, yep? And June Phos are important in the normal growth of some cells, and it's those cells, let's not make it up, but let's say they were important in the pancreas. It's the pancreatic cells that could have mutations in June and create pancreatic cancer, right? Like that. We've already seen that, right? That we see the PGF receptor pathway playing a role in that woman's skin cancer because it plays a normal role in skin cancer, in the skin cells, right? And so that's how we see different mutations in different tissues leading to cancer. And it's because, well, those genes and proteins play a role in that tissue. Like that, okay? Awesome. And so there's just that pathway again, and leading to, elk, great, and transcription. And so this would be an enhancer upstream of cyclins. That'd be one. There would be other genes that are going to activate it too, but, cyclin, great. Okay? And then just to show you, this is another transcription factor that we learned about in chapter 17. It's one of the ones that was at the end of the chapter, where it was just like the Wnt signaling pathway and the SMADs and the JAK-STAT. We kind of stacked them in. And this is just that Wnt signaling pathway, and it shows both the absence and the presence of Wnt in the same cell at the same time, just as a cartoon figure. So in the absence of Wnt, the transcription factor beta-catenin gets degraded. And its target genes or its response genes are inactive. In the presence of Wnt, the beta-catenin protein gets stabilized and can do its job, and it's to activate genes. And it has a lot of target genes. It includes Mick and cyclin. So the book wants to put this in chapter 19. But it makes sense to show you here, just to show you how the cellular response often includes transcription, and it includes transcription regulated by transcription vectors that we've learned some about. So elk, and then we're going to see Mick, and then this new one, June Phos, and then here's beta-catenin. And the book is just, has to show you more about beta-catenin because it got tucked in chapter 17. And beta-catenin in our normal health is way more significant than probably any other protein we've talked about. So they just got to, the other scientists in the world are like, my God, Jeff, our author, Jeff, we got to get beta-catenin in there a little bit. So, but see, it's satisfying here because we're understanding it. Oh, it's a transcription factor. It's dynamically regulated somehow. It's regulated. And it's going to regulate these things that are needed to get the light to turn green. Yep?
So when, one transcription factor regulates multiple gene expressions, are they usually, genes that have, related functions? Or can they be, just, completely?
Yeah. So remember this thing, when we did the clay model thing, we did a drawing of it in class, and I said, you got to make lots of dots for the MyoD, because when a transcription factor gets made, there are thousands of molecules in the cell, and that's just an important concept, because a transcription factor, because it has thousands of molecules in the cell, it can bind to every gene that has an enhancer for that transcription factor. And so we talk about the response genes or the target genes, and it can have a few or a bunch. Heat shock transcription factor has about 10 genes that activate, because there are about 10 genes with enhancers, heat shock response elements upstream. It just activates them all coordinately, like that, yep. And so, yeah, you just have to take it on a case-by-case basis. Estrogen receptor, just tons. I mean, just think about puberty, and the way puberty happens in vertebrate females is activating estrogen response genes. All of them have the estrogen receptor bound to them, bound to an estrogen response element, and they're just coordinately activated. And so, yeah, really typically, and so in a course like this, we're introducing it, and we don't say, and here's all the targets, but we'll say, here's a target of elk, that's satisfying to hear about. But there would be other targets of elk as well. And then when we pick a target, like cyclins, we're seeing, oh my gosh, right, on the test, you would say, okay, an upstream, or the regulatory region upstream of cyclin. And it would be that clay model video again. It would be like, there's the promoter, it's got some little clay proteins bound there, they're fine. And then here's an upstream enhancer, and it is binding elk. And then here's another upstream enhancer, and it is binding beta-catenin. And then here's another upstream enhancer, and it's binding June phos. And that just allows for versatility from different cells, right? Lots of different cells in our body get regulated in different ways, and so as a result, some important genes have multiple enhancers, so that they can be regulated by different transcription factors. There's also going to be an enhancer of mix. So let's just draw that idea, right? And we didn't start the course with that because you would have dropped it, right? And you'd be like, I'm taking my money over to SMCC. Like that, you'd just be like, cuz Dr. Champlin sucks. Oh my gosh. So here's the transcription unit, and it's for this cyclin. And we're going to make it, we're going to get the RNA, and then we're going to make cyclin protein. And remember, the cyclin protein is going to bind to CDK. CDK is a cyclin-dependent kinase. So that's something that's going to have to happen for us to turn the light green, because that's literally what the light is it's the CDK. To get that made, we have some options. We got options. And one is, well, we could get old elk in there, and it would be a phosphorylated elk, and get that thing to bind there and activate transcription. Or we could get June phos, and June phos is this heterodimer of June and phos. Let's just do this, and that popped up because it's going to pop up in chapter 20 because they're famous oncogenes, and so they must be vulnerable to mutation and getting activated. So we could get that going by June phos, or we've got one here for beta-catenin, so I'll put that up here. Here's beta-catenin, nicely stabilized in some cells. Beta-catenin. And then we'll make this one striped. So we're going to make a stripedy one here because it's MIC. So we'll do that, making stripes by making it first like this, and then be like, because MIC is just rowdy, so it's like a bumblebee that will sting you. So there you go. MIC. Right? And like I said, right, if that was the clay model at the start of the course, you'd be like, wham, right? But that's what it's like. And it's got all these guys, and you're just like Uncle Gene City, right? Like that. Upstream of this cell cycle regulator, right? Just trouble. We got to carefully regulate that. Yes, Hannah?
And then did you show previous, you saw that beta-catenin was binding to TCF, or just drawing TCF?
Yeah, let me just mention that, because
Like a dimer too as well?
it is a dimer, and we can understand that because we talked a little bit about this idea before. Beta-catenin does not have a DNA binding domain. So it's what's called a co-regulator. It's a transcription factor that's a co-regulator. And in this case, it's a co-activator. So it is a transcription factor, and it activates genes, but it doesn't bind the DNA directly, it binds onto another protein. And this is just a little satisfying thing to say for people like us who love transcription factors and are trying to decide which transcription factor to get tattooed on our right arm and which enhancer on our left arm, and just go out and be like, look at these guys binding together, right? That would be so cool. Like that. Because look at this one. This is a very good one. On our left arm, we have beta-catenin target gene DNA, and bound to it, in the absence of beta-catenin, is TCF. And TCF is a big-time repressor. So that gene is off. And beta-catenin gets stabilized in the presence of Wnt signaling factor and binds on the back of TCF and overcomes its repression and is an activator, right? So on our left arm, we've got, you could do this on the test. Just write it on your arm and walk up, hand in your thing and say, this is my answer for number 32. Oh! Right? Because it's TCF3 bound to the enhancer of some target genes, repressing them on your left arm. And then on your right arm is beta-catenin. And you bring it over, and it overcomes that repression and activates it. And that's what, that reason it's worth a tattoo is that's a molecular switch. It's a crisp molecular switch, like that. It's not a dimmer, right? It's not that we're over here and we dim the lights. We're just like, shut her down. Ooh, yeah. Right? And man, oh man, that is a satisfying thing. So detail that, we're kind of smoothly going over, right? Because we're really looking at that target gene, cyclin, and saying, look at all the different ways this can get regulated. But it's a sweet one. And so let me just show you this Mick. Yes, Godwin.
Go back to the drawing 1 second.
What?
He just wanted to go
The
back.
DNA, yeah.
Oh, go to the drawing of cyclin?
Yes.
Yeah, rainbow transcription factors. When I was in college, I took Latin, and the professor would go over here like this, and he'd do like this. James D. May. Love Latin! Like that, You guys have to
Love transcription.
I was sitting down. Who said that? Catherine, shut up.
Oh, I just have a quick question.
Yeah.
So is this like,
I'm sorry.
No,
Whoa, Rachel, I'm sorry. Oh my gosh. I got way too enthusiastic. Oh my gosh, everybody's coming in.
My students are just loving this.
I apologize. It's not going to happen again. Look what I drew up here, though. It's just.
It's beautiful.
I'm quiet. I'm quiet. Like, there's another instructor down there. I'm sorry. It sounded, yeah, I apologize. It sounded like things were. Well, that happened to you in college. Like somebody comes in and tackles me, and they're like, I don't know. All right. Yeah, no, I'm just shaking. Because it's just epic, you guys. Sorry, Catherine's asking a question. That'll help me set up. But that happened to me in college. That guy, James D. May, man, he was teaching us Latin. Shoot. God, that was bad. Right? Sorry. Shoot.
so my question, is this like any one of these can individually activate cyclin, or is this kind of like a pre-initiation complex where they all have to be like there?
Yeah, that's a good question. And the answer would be, it depends on the cell type, like that. So it would be, the answer would be that. It would be like some cases, just one of them can get the job done. Other cases, others are stuck, like that. One way to just think about that is, in some cases, TCF is bound there. Who's going to overcome that repression? Beta-catenin can. Could admit, I don't know. Maybe in some cells, maybe not others. TCF isn't always bound there. But yeah, there's a lot of interplay. It's important to remember that the bottom line is just, is the gene going to get transcribed or not? And it's a little bit like a kind of majority rules type of thing, where we got some repressors, some activators. And again, we're just getting a taste of the complexity of the switches, right? Because like we had a nice series of dominoes falling, and you're learning that, and you could represent that on the exam. And then just in this step, You know, it expands to kind of a lot of different stuff. Yep.
Yeah, so I guess these examples aren't just strictly enhancers, but there's more transcription factors.
The examples are really focused on the transcription factors because they always are. The transcription factor is the dynamic thing, and the enhancer is really just like a landing site
Right.
in the DNA. You gotta have it, it's an essential part of
But
the
you
story.
wouldn't have more than one enhancer.
You do. Every cell in our body right now has the cyclin gene with all those enhancers
Gotcha.
ready, because the DNA is not changing. So it's an essential part, but the enhancers are like a static parking site that's either occupied or not. Yep. Dang it. I wonder how bad I'll get in trouble. Probably some students are really feeling bad because they're like, I'm triggered. I'm triggered. I don't know what that yelling was. Yes, sorry.
No, you're good. So just to kind of again,
I suppose this is going out in the World Wide Web later today, so I'm probably like, do we have evidence, Your Honor? Sorry, Ellie, yes?
so you're saying they're like the parking spots, but there's not always gonna be a transcription factor
Right.
then that's really behind. It's just like maybe a couple or maybe one, as long as like
Yeah.
yeah.
Yeah,
Okay.
you know, for example, we saw today that in this cell, before the growth factor, quietly, before the growth factor was added to this cell, there was no FOS protein made. So there was no FOS protein bound to the cyclin enhancer, right? You know, so yeah, some cases, yeah, it just doesn't have the cell, but in other cases, maybe it has FOS, but not June, because it needs both, like that. I'm settling down. Personal timeouts. Yes, yeah, that's like in that Robin Williams movie where he gets in the Dead Poets Society. You ever see that? He gets fired. He's like, you're too enthusiastic of a teacher. Your students are not under control. All right, so let's just look at this, right? So we have this concept of, well, these transcription factors have multiple target genes because there's plenty of the molecules and they bind to all the enhancers. So this is just a list of some MYC target genes, okay? Just to show you another way of seeing this. So here's MYC target genes, response genes like that, and it says, yeah, we got cyclin genes, and remember, we just have to remember cyclin, we don't have to remember the different letters, but there are multiple cyclin genes, so that adds to the story, right? Right, we have, in this one step, multiple dominoes lined up side by side, right? It just gets complicated, but complicated in ways that we could walk through. MYC can activate these. Let's just see this. Here's a thing where in some situations, MYC can actually also activate or instead activate some inhibitors of CDKs. So this is a CDK activator. It could also inhibit some CDKs. So it just gets complicated like that. But bottom line for us is MYC could definitely activate cyclin and get things going. It can actually activate some other things that would be like that school patrol going up. No, not going yet, right? So just gets to, get into the weeds like that, right? So in that context, here's the CDK that's at start. It's actually multiple CDKs at start, but here's the one we're going to learn about, and we're just going to call it CDK, cyclin-dependent kinase. And there's that cyclin that we've been talking about that it needs to be active. And what we're going to learn, and our book emphasizes just, and it's more than this in real life, but just 3 ways to activate, to regulate that CDK. Okay, and I'll just skip ahead to this list here. So examples of CDK regulation at start. One is, you gotta have cyclins, they're cyclin-dependent kinases. And we usually gotta make them. So usually gotta have transcription vectors bind the cyclin genes and get them made. 2, it can be regulated by phosphorylation. That's complicated because sometimes it helps activate the CDK, sometimes it helps inhibit it. We'll talk a little bit about that. Then 3, there are some inhibitory proteins that could stop it, like the school patrol. So those 3, and they're represented here too, visual. So here's the CDK, that's the enzyme that is the light that is red and it's activated, it's green. And what is regulating it could be cyclin, need for cyclin, could be that you have some phosphorylation sites that could activate it or repress it, those are regulating it. Could be an inhibitory protein. Like that, those 3. And we'll learn about those, and we're going to pause here for a concept chat, and we're not going to yell. We're just going to gently just go through this. And so before we stop for a concept chat, let me just say that, see, this is going to turn the light green, so we need to activate it. Cyclin is an activator, and in some cells we need to make cyclin, and in those cells, it's a transcription factor that's being regulated. And so that would be activating the CDK. And so it's pretty reasonable that upstream of that cyclin, there are several oncogenes because we could accidentally make it when we shouldn't. The woman with skin cancer, that's her problem. ELK gets activated on that RAS pathway, we make cyclin. So think of this one, it's an activator, cyclin-dependent kinase, and it's activator, and stuff that's regulating upstream, like those transcription factors in the signaling pathway, those can have oncogenes. Now, just as an introduction, because again, this is just a taste of SARP, this is an inhibitor. So this is never an oncogene. It does the opposite. Whatever's regulating this upstream of this is not an oncogene, because this is an inhibitor of the CDKs. And these proteins, and we'll learn about that in chapter 20, but just to say it now, because it's really satisfying to know, these proteins are referred to as tumor suppressors. Their normal function is an inhibition of the cell cycle, and so they could help suppress tumor growth. So let me just write that out here. You know, everything here we talked about and I was yelling about, and God was like, I like those different colors on that drawing, I want to see those again. Those are oncogenes, and oncogenes can get people really excited. Tumor suppressors do the opposite, and
Are all of those transcription factors oncogenes? Like
all the ones in our colored drawing, yep. Yep, in our tattoos, yeah, those are, yep, that's what we got tattooed on our arms for, tumor suppressors. Dang it. Those professors that came in, it's funny too, because they've been a professor here a long time, and all of them laugh, and like when they see me, they're, hey, Dave, and I like as soon as I pass by them, they're like, what a loser. You know, it's like, you know, he's no good. How do you get a job?
I
So
doubt Rachel says that.
Rachel does. I can tell you sometime why Rachel says that about me too. It's sad. Tumor suppressors, that's their normal function. So that's those inhibitors. So we'll learn some about that. And again, it adds complexity to the start because the school patrol is like a tumor suppressor, right? Now, those can get mutated, and in many cancers, we have mutations in tumor suppressors. And those are common because when you make a random mutation, a lot of times you damage the function. And you can damage the function of tumor suppressors, and then they're just not there to help put on the brakes, right? The school patrol has a hangover, and they're just like, good luck, kids, you're on your own, you know, and you're like, the intersection is quite dangerous today, right? Nobody's gonna get hurt, maybe, but you don't have that tumor suppressor to really provide the extra help. And so it's just a dangerous game. So when you have a mutation in tumor suppressor, it's not uncommon because you just have to wreck them by random mutation, but it doesn't cause unregulated growth. It just causes a failure in a check and balance situation like that. Yep.
Having an overactive tumor suppressor, would that cause a difficulty in growth and wound healing?
I would say yes, yeah. Just, kind of theoretical or, in principle, yeah, yep,
And
yeah.
the CDK is only activated by the trimerization of CK1 and cyclins.
Sorry, I was still thinking about that yelling. So what's activated by?
On the figure, CDK is only phosphorylated when trimerized with
No,
CK1.
each of these three things can happen separate from the others.
Okay.
Separate pathways to regulation.
So what kind of happens first?
It varies.
It varies.
Yep. It's just like this thing where you have three things you need to get done before you leave the house, and the order doesn't matter. You're just gonna have to get all three of those done, and they're separate stories, yep?
But CDK is only activated with CK, is that 1 or an L? CK1.
Orange neck.
Oh, CK1.
CK1 and cyclin.
When this is present, it's like that skill patrol where it's like it overwhelmed everything. It's a trump card like that, where it's like nobody's moving forward,
The
no matter what.
repressor and the activator work together to activate.
No.
No.
Lots of times the repressor is just not there, right?
Okay.
And so all we need to do is activate this thing. Think of the repressor as this thing that can be like the school patrol, where it's just like this extra separate thing, and it can just say, nobody's going forward. It doesn't matter if you have cyclin, you're not going forward.
So
It
like-
doesn't matter if you have the right phosphorylation, you're not going forward.
And similar to that, if there's no phosphorylation, CDK is not gonna be activated.
That's not as crisp as these inhibitors,
Understood.
but generally that's the
Similar,
concept,
similar.
yep. Another thing to say is that you're not going forward without cyclin. You gotta have that. But people would say that these inhibitors is because the famous examples are where it's like this trump card thing, where it's just like, it doesn't matter the status of anything else. If I'm here, it's stopped.
Okay, that's what I wanted.
Like that?
Yeah.
And that it's like that skill patrol where it doesn't matter if the light is green or red, really doesn't matter if there are children present. If the school patrol's out in the intersection, nothing else matters. They've got that red little flag. And I don't know if you guys know what school patrol is, but it's at a school where they have a person walks out with a little flag in the intersection so the kids can more safely cross. When that person's in the intersection, none of the cars can move, period. Doesn't matter. Don't have to check in on anything else. Yeah, so just that, yeah? No, but Eva still wants to follow up.
Sorry,
Yep.
yeah, the figure is just slightly confusing me because it looks like CDK is only activated when CK1 and cyclins are present.
Because of how it's kind of stuck together?
Right, so
Yeah.
you're saying regardless of whether or not cyclins are present, CDK isn't going to get activated if CK1 is around, because
Yes.
that's a stop all.
Mhm.
So CDK can become phosphorylated regardless of everything though.
Mhm. Yes, the phosphorylation status can change, but if this inhibitor is present, nothing else matters.
So that
Okay.
CDK in that picture is inactive.
Yes.
Because of the CK.
And if it were active, CK1 would just
Not be there.
around.
Yep. Yeah, and yes, and then there was another question up here, and then Godwin. Or was there a question up here? Nope. Okay, Godwin.
So, I have two questions. So the first one is, the three regulation of CDK.
Yeah.
So all of them are in this picture, right?
Yes.
You have the
Mhm.
inhibitors, CDK,
Yeah, right.
and you have the
Yep.
seven.
So that list is
Yes.
that same visual. Yep.
Yeah, so I think what we're trying to understand is the phosphorylation part, because we get the inhibitor, right? So inhibitor stop everything, CDK activates cyclin
Yeah.
activity. So the phosphorylation part is where.
Yeah, and we're not talking about that yet, and we won't talk about it very much because it's actually the most complicated part.
Yeah.
Because it can be both activating and inhibiting depending on the phosphorylation. So just say there are these three parts, and this is how we would say it right now, is that, cyclins, phosphorylation, and these inhibitory proteins each play a role. Yep? Yeah?
The CKI, you said it was tumor repressor?
Uh-huh. Yep, yeah, and I put the names of the CKI, I've forgotten what the book is calling CKI. It, I think is it says cyclin inhibitor?
Yeah, I saw it somewhere.
Yeah, I think it, because it's, I don't think I think it's kind of like a like some way of trying to come up with a general name.
Wait, so is that what?
Yep.
So sorry, is that CKI or CK1?
CKI, I believe. But again, I don't even remember what that's for, because I just don't think they're commonly called that. The famous thing to call this thing, the most famous thing is they're called small inhibitory proteins. And that's a general name for a set of specific proteins. And here's two. I think we're gonna learn the names of two. So see, binding of inhibitory proteins, let's just get specific. That CKI, I don't know what that is. It's an inhibitory thing, but P21 is an inhibitory protein that we'll learn about, and P27. And those will be in chapter 19. Yep,
When you say, is that page 21?
Nope.
Okay.
that's a good question. So, this is a way of naming proteins when the people didn't know what they were studying them, they didn't know what they should call them, and so they would run a gel just to see the protein on gel. And the one thing they'd learn about them is the size, the length of the protein, or the molecular mass. And so what this is saying is really just an old-time name that stuck. It stands for protein that is 21,000 daltons in size. So one of the reasons those names stuck is because that's pretty small for a protein. And just, coincidentally, the inhibitory proteins in this story that were discovered mostly are just surprisingly small proteins. So, like I said, most people call this group small inhibitory proteins because there are several, and they tend to go by the names P27. There's one P53, like that. Well, no, not P53, but it got its name because it's a famous tumor suppressor, but it's not, it doesn't bind to this directly. So erase that, just go with P21, P27. And yeah,
57.
yeah. Huh?
And also 57.
P57 is one that's listed in the book? Okay. Yes, Ellie.
So would you say like you need cyclin present phosphorylation and no inhibitory proteins to be able to make CDK or activate CDK?
I think the way to say it would be that this is called a cyclin-dependent kinase. That's the protein that we need to regulate. And it has to have cyclins, cyclin-dependent.
Does it have to have this other phosphorylation in it?
Not always.
Necessarily.
Yeah,
Okay.
yeah. So start with that like we did in class. Gotta make those cyclins, and a lot of times that's enough. The CDK is always there, it's just not active. You're gonna activate it. Just getting cyclin made, that's enough. Then, and so that's satisfying because we got the enhancers on that gene. It's like, okay, if you get that made, there's a good chance you'll roll with it. Not if it's decided to make a small inhibitory protein, no. Right? And then this is additional regulation that could sometimes help activate it, sometimes help repress it, sometimes it isn't even needed. Like that, yep. It just, because of that, the phosphorylation becomes complicated, and we spend the least amount of time on it, and the most amount of time on these others, partly because they're super famous, in the stories, and partly they fit into our course learning. Whereas the phosphorylation stories they just are variable things that are just a big stack of biochemistry that's different and not so easily memorized or kind of building a framework of what is happening. Yep, so we'll just talk about them a little bit, but we want them on our list. Yep, and we're gonna run out of time for concept chat, so we should just have a couple more questions if everybody has questions. I apologize to all of you about yelling. Well, you were in here. These other people just heard it at a distance and they thought, wow, that's a serious professor take down. Like, who's taking who down? Is that a professor taking a student down or a student taking a professor down? I need a drink of water. Yes, Godwin?
So when you do the DNA, right?
Yeah.
All of those things were binding to the enhancer, right?
Multiple enhancers.
They
Yep.
were all on the same DNA?
Yep. Yeah. So go back to the clay model video, and we said, oh, MyoD binds to myogenin and activates it, because it binds to an enhancer. Now just imagine that myogenin has multiple enhancers with different transcription factors that could bind to each and regulate it. That's what's going on. Yep.
So we have the CDK, which is like, as usually the low pathway. That's just a protein, not an enzyme.
It is an enzyme because it's a kinase, cyclin-dependent kinase, and kinases are always enzymes. Their function is to grab a substrate and phosphorylate it.
And when you say CDK regulation, or all this stuff that you're looking at, are you saying there's this gene that also regulates CDK? These are all the things that you need in order to regulate.
Sorry, what? Yeah, no, I just didn't hear you. Yep.
So, in order to, we have things regulating CDK, as you can tell here, and is this on a gene? Like, would this have to all come together on
Nope.
no?
Nope. Now, this is not a story of gene expression. This is these
That's
are
like
proteins.
the actual enzyme right there.
These are proteins just floating around in the cell. Yep. Yeah. All right. Okay. Cool. We got to stop. Obviously, I'll have to go